Hello, everyone. Welcome to today's webinar. As folks are getting logged in today, I'd love to tell you about some upcoming events that we have going on. Next, on Wednesday, May 17th, we're going to have an education and research webinar on ergonomics with the impact of precarious work and psychosocial stress on the physical and mental health of California janitors. And that will be with Melissa Achterman, MS, CPE. On Wednesday, May 24th, we will have a webinar on supporting healthy work, law, and regulation with Walter M. Stella, JD, and the California Labor Lab. Our next ERC, our industrial hygiene webinar, will be on Tuesday, May 13th, and that will be on sampling methods for personal exposure to herbicides with Patrick O'Shaughnessy, PhD, CIH, FAIHA. For more about these upcoming events, you're welcome to visit us online at coeh@berkeley.edu/about-CE. And on behalf of education and research centers throughout the country, it's my pleasure to present the Industrial Hygiene Webinar Series. This is a collaborative effort on behalf of each ERC's continuing education program and aspires to provide access to current research supported through NIOSH ERC programs. We appreciate you joining us today. Today's webinar, Occupations at Risk for Vector-Borne Diseases, is brought to you by Wayne T. Sanderson, PhD, CIH, and the Central Appalachian Regional Education and Research Center. Few housekeeping items before we get started today. You will be muted during the presentation. If you'd like to ask a question, please enter it into the online Q&A, and we'll save time at the end of the presentation to address as many questions as possible. This presentation is being recorded and will be made available on YouTube, and we'll send out a link in your follow-up email after the webinar. All participants who logged in with their registration email for the full live presentation today will receive an email tomorrow with a link to the evaluation form that will qualify you for a certificate of completion with 1 continuing education contact hour. Once the evaluation is completed, you'll be able to access and print your certificate. Without further ado, it's my pleasure to welcome our presenter today, Dr. Wayne T. Sanderson. Dr. Sanderson is a professor in the Biosystems and Agricultural Engineering Department in the College of Agricultural, Food, and the Environment at the University of Kentucky. He is also director of the Central Appalachian Regional Education and Research Center and director of the Southeast Center for Agricultural Health and Injury Prevention. Dr. Sanderson conducts research on respiratory diseases, cancers, infectious diseases, injuries, chronic diseases, and adverse birth outcomes associated with a wide variety of occupational and environmental exposures. Thank you so much for being here today. We're looking forward to your talk.
Thanks a lot. I'm grateful for Northern California ERC hosting this seminar series and delighted to be a part of it. I think I have one of the best jobs in the world being an ERC director, because those of us involved in ERC activities, we get to help young people decide their way in a career, help them along, mentor them, and most importantly, we get to learn from them. And so this presentation today, I have to give 99% of the credit to my students, in particular, Dr. Anna Pasternak, who just graduated with her PhD in entomology last week, but she was one of our ERC trainees with a focus on occupational diseases related to vectors, in particular ticks. And I hope this isn't too elementary a talk. It seems that a lot of people really don't know that much about ticks and tick-borne diseases. And you may be asking yourself, so what does this have to do with occupational related diseases? And I'll get to that toward the end of the talk, I think. But to begin with, I kind of want to just give some basic background information. Maybe you didn't know that much about ticks. I didn't. I learned largely from my work with my students, Anna in particular, but also others. And we actually have a fairly robust program in our ERC and Ag Center on vector-borne diseases affecting particularly the agricultural community, but other occupations as well. So some background, just a bit, and why I think maybe people don't know that much about ticks. I do surveys when occasionally, I can't do this on a webinar like this with over 100 people, but in a classroom setting, commonly I will ask people who has never had a tick on them. Now, a farm boy from Western Kentucky, growing up in the woods and the fields, I don't think there was a day went by. I didn't, in the summertime in particular, I didn't have a tick on me. But it's amazing that there are people out there who say they have never seen a tick on their body. Maybe not. Ticks can be really, really hard to see. In fact, most people who develop tick-borne diseases would say they don't recall seeing the tick, much less getting bitten by the tick. But ticks are all over the world. There's over 800 species. I'm really going to focus on three key tick families, I guess you could say, or tick orders that we can see here, largely in the United States, particularly in the East and the Midwest. So ticks reproduce by internal fertilization. The males transfer a spermatophore to the females, but there are some females that can reproduce with parthenogenesis. And as I was telling Michelle, if this was a regular classroom, I'd call on somebody by name and say, what is parthenogenesis? Well, hard for me to do that in this kind of format, but parthenogenesis is when females are able to reproduce, are able to lay eggs that are fertilized with asexually. Basically, it's a cloning kind of operation. Now they tend to basically be almost exclusively female progeny, but there are some ticks, and particularly the Asian longhorn tick, what I'm going to talk about later in the talk, can do that. Not all ticks can, though. Certainly not all animals can do that, but not all ticks can do that as well. But all ticks go through 4 life stages: egg, larva, nymph, and adult. And this is why I said that people may not have even known they were ever bitten by a tick. Look at that tiny little tick on that person's fingernail. That's a larva. After they hatch from the egg, they really are that small. They're about the size of a grain of sand. You can see them with the naked eye, but they are very, very small. They're only 6-legged, and they do take a blood meal. But typically they don't transmit diseases because they just came out of the egg. They haven't had a chance to bite anybody and get infected with some organism. So they get a blood meal. They take a blood meal from some animal or from a human, and then they can molt and develop into the next stage, which is a nymph. And the nymph you can see on this person's hand, a little bit bigger, about the size of maybe a poppy seed. Now they have the 8 legs that are associated with the tick family. They pose the greatest threat to disease. The reason is because they're so small, we can't see them, and they've already had at least 1 blood meal. So it's possible they may have been infected with some agent, some organism that can cause disease. And then adults is what we're probably more commonly used to seeing. Is they're about the size of maybe an apple seed or so, and they live about a year or 2, and typically after they mate, they die. Now, some ticks a few will attach to a host, and that's where they spend all their life. Some ticks will attach to a host as immature ticks, and once they're adult, they jump off and move through the woods and the grass and live and bite somebody else, some other host for as an adult. But most species, they will find 3 hosts during their life cycle. Okay, well, that means that if they've bitten some host that has an organism, then they can certainly transmit that on down the line to someone else. Just a little background information, if your biology is a little rusty, just to talk about what I mean by reservoir hosts. These are animals which may harbor the infectious agent. So if the tick bites them, through the blood meal is able to bring that agent into their body. And then as they go on and they bite another animal, they can transmit that to the next animal. And so the vector, the tick, serves as the mode of transmission, and the reservoir host can have that agent out there in the environment. Also, a term that you may not be familiar with is dead-end host. People are dead-end hosts. If you get bit by a tick and you have that disease, the chance that your family is going to get bit by that tick from you and spread that disease to another person in the home. That's really not going to happen. I mean, we tend to be dead in host, and our pets tend to be dead in host, which is, fortuitous, but still, if we get the disease, not so much. It turns out that I didn't know this, but tick-borne diseases are the second leading cause of arthropod diseases worldwide. Now, of course, mosquitoes worldwide are the number one. You've got malaria is still very, very much with us, and we still have yellow fever. We still have Zika and dengue and chikungunya. All are mosquito-borne diseases. But in the United States, the number one vector-borne, the main vector-borne disease is tick-borne diseases. And these are notifiable diseases. And then if you know what notifiable diseases are, that's a passive surveillance system in which we rely on the medical community and their associated laboratories that once a tick-borne disease is identified in a patient, that gets reported to the state departments for public health and eventually gets reported to the CDC. And so that's the system we have. But the problem, and this is where our research came in, is we believe in the literature and folks doing this kind of work that underreporting of tick-borne disease is a major problem. And the reason we think that is something like Lyme disease, which is one of the more common, it is the most common tick-borne disease in the United States. Maybe 30,000 Lyme disease cases a year or so get reported to the CDC, and yet many more people are getting treated for the disease. So we think that at least the physicians are suspecting that the people had Lyme disease and treated them for it just in case, even though they may not have ultimately reported that as a Lyme disease case. The map on the right shows all the major tick-borne diseases lumped together, and it shows their geographic distribution. As I said earlier, this is primarily a problem of the Northeast and the Midwest, and moving into the Southeast. Here we are poised for our research and our continued activities, trying to determine what diseases, emerging diseases, are coming into our region, and how that might affect occupations in particular through the ERC, but the public in general. Having said that, there still are tick-borne diseases throughout the United States. In fact, was a personal note, I actually was a Rocky Mountain spotted fever case way back in 1980, very, very long time ago. And I got it in Arizona. I think I was one of 12 cases in Arizona that year. Even though it's called Rocky Mountain spotted fever, the predominant area, the areas of highest incidence for Rocky Mountain spotted fever are in the east. So why do we think tick-borne diseases are underreported, or what might be the cause of that? Well, we think it's possibly a lack of education in our healthcare community, a lack of education in the public in general. And in fact, one of the things I said that's so great about ERCs is we're so interdisciplinary. Before we started the webinar, Michelle was telling me there's a lot of industrial hygienists on this talk. And industrial hygienists, you've got a lot of roles to play in trying to help identify and prevent tick-borne diseases. We got safety professionals, we got medical people, we got nurses, engineers, ergonomists. We are very, very interdisciplinary, and yet, how much instruction have you really had on vector-borne diseases? Even in medical school and nursing training, I hear that it's not touched on to a significant degree unless you go into a residency that focuses on infectious diseases, but in general. And when you think about places where Kentucky is a very rural state, we don't really have a lot of infectious disease physicians, people with that focus out there. And we're hearing anecdotal cases of people who developed some symptoms, and it lingered for a long time before they were finally diagnosed with a tick-borne disease. Also, the cost for testing and the complexity of those tests are difficult. And so not every lab and not every healthcare facility is going to have that ability. Also, tick-borne diseases mimic many other diseases. So it's quite common to be diagnosed with some other more common disease than possibly a tick-borne disease. And of course, a big concern to us is with the climate change. We are seeing the emergence of ticks that we had not seen before, and coming into our region, coming into various regions. And so that's one of the focuses of our research to date. And I'll talk to you about how we're building upon that. Now, the most common tick-borne diseases in the United States, but this is early research. We just this year are starting our more intervention work, but this is the background information or background research that was done. There were really 3 basic diseases, tick-borne diseases in our state, but that's true of the United States in general. And so I'm going to talk about spotted fever, rickettsiosis, Rocky Mountain spotted fever being one of those, but CDC's lumped the rickettsial diseases together under SFR, ehrlichiosis, and then Lyme disease. And you can see those numbers on the right. As I said earlier, by far, Lyme disease is the most common that we're seeing in the United States. But you might see these jumps in numbers too. And we're seeing an increase in tick-borne diseases pretty much across the board. And so what we want to know is we want to come up with some, the objectives of our research was to determine the distribution, abundance, and seasonality of those tick vectors, and then to determine whether agents were in them. So what kinds of ticks we're finding, where we're finding them, and are they infected with the infectious agents? That was the purpose of this research. And then where we're going with our future research is trying to focus on, well, if it's here, if it's in our region and it's in the ticks, are we seeing it in the people? And if we're not seeing it in the people, have they been infected and not being diagnosed, or are we just not seeing it in the people quite yet? But it's something to worry about, and that's our direction. I'm going to go through these a little quickly because I want to give some time for questions, but not too awfully fast. I want to go slow enough that you kind of get a flavor for it. Lyme disease is the most common tick-borne disease in the United States, and it gets its name from a valley in Connecticut. That's where it started some decades ago, but it is spread all down the East Coast and also high pockets there in the upper Midwest. The active agent that causes the disease is Borrelia burgdorferi. And it is this agent can be in white-footed mice, small mammals, birds, even reptiles can harbor this agent. It's not usually fatal. In fact, that's not the main concern with Lyme disease, but the main concern is the neurological symptoms. Is if it's left untreated, people will develop some joint pain and neurological problems on down the road. So that's the concern, is these, and it becomes chronic, long term, and then there's not much we can do to other than treat the symptoms. That the agent is already set up housekeeping and causing problems. You may have heard of a characteristic of Lyme disease is it causes the bullseye kind of rash, and that upper left picture, that's classic. If you see that, you're probably seeing Borrelia infection. That's a very common, but not always. It doesn't necessarily mean that if you don't see that exact rash pattern, that the person did not have or does not have Lyme disease, because the other patterns can be exhibited. What we do tend to think, though, is more common if you see a rash, a reaction for a long period of time. I mean, if you've been bitten by a tick, it is not uncommon to have basically, hey, there's a little spot, but no big deal. Or you have a tick and you have a red area around it. But if you have a tick bite and there's a clear lesion that lasts for a period of time, we have even greater concern that maybe there was some agent. Your immune system may be able to fight it off, and then again, it may not. It depends on your own individual constitution and the amount of agent that was injected into you. As I said, Lyme disease is at epidemic stages. We pretty well are seeing it spread throughout the eastern part of the United States, well established in certain parts. And the main tick that carries it, in fact, this may be what you think of when you see a tick, is this kind of black body with a light brown or lighter brown, still dark brown, but a lighter brown abdomen. Ixodes scapularis is the primary tick, the black-legged tick or the deer tick that carries it. And there hasn't been a lot of studies of this, particularly in the Southeast. And so that was one of the things we wanted to determine is Ixodes here in high concentrations, and if it is, has it been infected with Borrelia? Now I'm going to take a sidebar here because I'm not sure that any of you have been exposed to this. I was not until I started doing this work a few years ago with, as I said, with my students. And that's a picture up there with Anna. And this is highly sophisticated. The industrial hygienists are really going to perk up on how complex and sophisticated tick sampling is. But what we do is we take a cloth that's relatively 1 meter by 1 meter, typically a muslin or canvas cloth. We hook a rope to it and we drag it through the woods or the grass. And then when we're all done, we get down, and I have to have a magnifying glass, particularly with the little nymphs, to see them. But younger eyes can maybe pick these out. But that's basically what we do. There's Anna and her team walking through the woods and the pastures, surveying. We went all across Kentucky. Basically, I think every county was surveyed during our research the last 2 or 3 years. I think there's even a picture of me in the upper column there, trying to figure out how to use this complex piece of equipment. I have some issues with it. There's a number of things can happen. How characteristic is the sampling of the true tick population? I mean, we typically go out into public areas like state parks or areas that national forest and things like that. But we also ask permission from farmers and landowners if we can go visit their farms and walk around. Well, how does that really, if we survey 2 or 3 farms or woods in a county, how does that really represent the entire tick population? But that's what we do, is we pull the muslin cloth through the forest. We count the number of steps, and that becomes our sampling survey. I think maybe it would probably be better to just keep sampling and sampling, because in truth, I pulled the muslin cloth for 10 meters or 10 steps and counted 29 ticks. And then I pulled the muslin for 500 meters and not collected a single tick. Well, there's going to be a lot of variation in where ticks are, what they have, the time of day that I sample. Ticks aren't stupid. They don't like it when it's 90 degrees outside and no shade. So if I'm sampling at 2 o'clock in the afternoon instead of 8 o'clock in the morning, I may not pick up as many ticks. If I happen to be sampling where a deer spent the night or a bunch of little mice had a home, then I'm likely to pick up a few more ticks than if I'm sampling in a soybean field where nobody was sleeping that night. So the sampling process has some issues and questions that I think we'd like to explore further. Anna also had a passive surveillance activity where people would send male ticks into her, and she had a form that would fill out, and she got a number of ticks through the passive surveillance system as well, and of course would document the conditions. And then what we can do, with the advent of PCR, what an amazing tool is, we are able to, from these ticks. Now we have to group the ticks together. We don't usually get enough DNA from one single tick, but we might group them into batches of 10 or 20 ticks together. And then we would extract the DNA and look for the pathogens. We look for Borrelia for a particular gene target that it possesses, to try and determine if those ticks, we would identify the type of tick and lump them together as a group of the same type, but then we would try and determine which batches were positive, and then talk more about that here in a minute. It turns out that for Ixodes, found it year-round, even in the winter months. So people say, hey, you can't get tick bit in the winter. That's not really true. It kind of depends on who you are, where you live, what you do for a living, and what kind of ticks we're talking about. So Ixodes is extremely durable tick that can even come out in the winter months. And these are the counties in which we found Ixodes, and in 16 of these counties we found Borrelia, and which means a minimal infection rate of about almost 5%. Now, what I mean by infection rate, remember I said it's called the minimal infection rate. I would take the batches of ticks that were positive and then divide that by the total number of ticks that we sampled throughout all the batches, and then multiply that by 1,000. That's the classical, simplest way of coming up with what's called a minimal infection rate. And that gives us an idea of the relative level of Borrelia circulating in the Ixodes population. So that's the way that is done. So some summary points back to my talk on the diseases is Ixodes, the black-legged tick, was active in each month. So caution, year-round. Adults may be a little more common, October through June. The immatures emerge in May and may be active more in the summer, but you can find the adults even in the cold, cooler months. This species was collected from about half our counties. We have 120. We found it in, I think, 58 or 68 counties, whatever I said earlier. Now, not high infection rate, but it's clearly here. Borrelia is clearly here in Kentucky. So we can't tell people that, hey, you got bit by a tick. What did it look like? Did it look like the deer tick? Don't worry about it. We don't have Lyme disease in Kentucky. And we pretty well knew that we just didn't know the level of degree. The next tick I'm going to talk about, this is a really interesting tick. We did not have this in very high concentrations, very high populations some years ago, but Ambliona americana are what's called the Lone Star tick. And the reason is, this happens to be a female, on that leaf there is a female Lone Star tick. It has a white spot on her back. Doesn't look like a star to me, but got the name the Lone Star tick because of that. Now, interestingly, the male doesn't have that big white dot. It has more like 3 little white dots on closer to the end of its abdomen there. But the Lone Star tick is well established in the southeastern part of the United States. But with climate change, we're seeing it dramatically expanding to the north and to the west. And so it has become one of the most common ticks to bite people in the southeastern part of the United States in our region. And why we worry about this tick, there's a couple of things. It causes a lot of diseases. It could carry the bourbon virus, ehrlichiosis. We are one of the high ehrlichiosis states here in Kentucky. Heartland virus, tularemia, another tick-borne disease. Stari, and a new disease I'm going to talk more toward the end called red meat allergy. If you're not familiar with that, that is an exploding disease here in the southeastern part of the United States. As I said, I talk more about that here in a minute. The other thing about the amblyoma, it'll chase you. It'll come after you. Most ticks, when you think about getting a tick on you, it's because you were in the grass or you were walking through the woods and you swiped up against some brush, or you got under a tree and shook that tree and the ticks dropped down on you. I've actually been sitting there taking a break, drinking my water, and see one of these guys come after me. And I said, I don't know. I asked Anna, what is this thing doing? She says, well, I don't know if it's the carbon dioxide we emit or some pheromone or what is our perspiration, but they will actually come after you. And she says, I have actually had one chase me. I try to move away and it just keeps coming. So yeah, kind of an aggressive little tick. As I mentioned, ehrlichiosis is an emerging disease, and we're one of the the high states, it attacks the white blood cells. Infection can be quite mild, but if left untreated, it can be quite troublesome. We have had a number of case reports in our region where people developed the disease. It was misdiagnosed or not diagnosed for quite some time until they actually sought treatment, sought diagnosis somewhere else besides the rural area in which they worked. And so this tick that there hasn't been a lot of studies on it. It's not nearly as common as Lyme disease, but as I said, whereas we have a fairly low incidence of around 10 cases nationwide per million persons. Here in Kentucky, we're up around 17 cases per million people, and we're seeing a fairly high positivity rate in dogs as well. This one doesn't tend to be found in the winter months. Much between May and September is when it's most active. So that's a plus. We don't see it as much in the winter. But essentially, we found this tick in I would say every county. I think it's the sampling design. When we didn't find it, as I said, we have 120 counties, and we found it in 115. I think it was just chance sampling. I really think that amblyoma is throughout our state and is here to stay, well established. And if ehrlichiosis begins to be seen more prevalently in these ticks, we've got a real problem. So that's the concern with that one. The last disease is one that you probably have heard most associated with ticks. It is the one with the highest fatality rate. As I said, CDC is lumped in a group of illnesses from the rickettsial diseases is now called spotted fever rickettsiosis. That's relatively new, relatively recent change. This disease has a 25% fatality rate. Before the advent of antibiotics, that's pretty high. 25% of the people get this can potentially die. And of course, it's dramatically decreased with the use of antibiotics. They're quite effective. We haven't been seeing, I don't think, at least I'm unaware of us seeing antibiotic resistant spotted fever rickettsial diseases. But if left untreated, within a couple of weeks, you still have that fatality rate that you would have had before antibiotics. So it's a big concern to educate people about this. The symptoms fairly mild at first, but it does have that red petechial rash that's a telling sign. So the concern is, and as I said, people may not even know they got bitten by the tick and yet develop this disease. And so a number of different ticks can spread rickettsial diseases, but dermacentor is the main one that the American dog tick is its common name, and it is throughout the eastern part, midwestern part of the United States. It's a very common tick found both in brush and open fields. However, fortunately, even though it's an extremely common tick, we only are really finding less than 1% of the ticks being infected with rickettsial diseases. So that is somewhat of a relief, but it's certainly something that we want to continue to watch, and something that we worry about from the misdiagnosis standpoint. And as I said, I'm going to jump through these a little quickly because these are other rickettsial diseases that are associated with it. We found a dermacentor mainly in the summer months, the warmer months, in almost all the counties, almost 99 of our 120 counties where we found it. So the summary points on Rocky Mountain spotted fever are that it's mainly a warm weather tick. The major activity for this tick is May and June, particularly when farmers are out there planting their fields. It may be one of the most common ticks that they would find. it's widespread, and Rocky Mountain spotted fever, fortunately, the cases are relatively few, but it is a concern because of the issues. So from Anna's research, from what we've done so far, from our tick surveillance, trying to answer that question, what ticks are here? What are the relative proportions in the population? And are we finding the disease agents in the ticks? Lone Star tick was the most common, most widespread, and it's a vector for ehrlichiosis, which is a concern, and it's mainly found in the summer months. The dog tick was the second most abundant, with peak activity again in May and June. Ixodes, the black-legged tick, was third most abundant, and it appears basically active in each month. So no question the ticks are here, and no question that we see the disease organisms in some of them. So we need to do a better job of educating people about this. So what are some of the things that we're doing? Over here on the left, I think I have some, is we're putting out, this was a CDC idea we stole, and I think I can hold this up to the camera. This happens to not be a tick, but a bed bug. Whoops, oh, I got to put it here. There's my camera. Is we've developed these baseball-like trading cards for various vector-borne diseases. We've been taking these to talks at schools. To you can see us down here. We had a display at the FFA convention in Indianapolis this past fall. We tried to go to FFA. It's not that far from here. It's at University of Kentucky. About 60,000 students, FFA members, come to that convention, and we tried to set up an educational booth on a wide variety of things: machinery safety, stress and mental health issues, water quality issues. But the last couple of years with Anna and her team came up with this idea. You might see her T-shirt there. She has a tick. It says Tick Talk. I don't even know what TikTok is, but Tick Talk was catchy, and so she had a demonstration there to educate students about vector-borne diseases in general. She did have bed bugs there, and cockroaches, and mosquitoes, and ticks. But one of the things we think we need to do is be educating people about this. Now, I promised you that I was going to tie all this back into what does this have to do with occupational safety and health. Well, it turns out that there are occupations which have intense tick and other vector pressure. And you can imagine who those would be. I mean, as I said, we have an ag center here, and we work with farmers, and they have clearly one of the greatest risk for tick-borne diseases. I mean, every farmer would tell us. So we say, "Oh, did you get bit by a tick last year?" And they'd say, "How many times?" Now, another group are loggers. It turns out here in Central Appalachia that there's a number of logging companies. We have a fantastic hardwood logging industry. And most of these companies are fairly small. Doesn't take much to start a logging company. If you got a chainsaw and some equipment, a lot of these are family-run businesses with 4 or 5 people running the company. So you can imagine there's no industrial hygienist or safety director out there watching these people. They do have to come in and take training. We have each state will provide master logger training, and health and safety is part of that. But also forestry and fishery people who basically spend a lot of time in the woods and the fields, are at risk of being exposed to these agents, to these ticks, these vectors, as well as the potential diseases that they may be carrying. As I said when I started off, being associated with an ERC, particularly the director, you get to learn a lot of stuff. And so I'm going to highlight here my last bit of the talk. Paul Bellamy came to us to our ERC as a trainee with an entomology undergrad degree, but he wanted to focus more on occupational environmental health. And so he came and wanted to be a trainee, and we said, well, we haven't had an entomology student, we'll bring you in. Well, after a year, it comes time to do his capstone, his thesis, right? What's he want to do? Well, he told me that there was a disease called red meat allergy, and he wanted to study that, and he wanted to study it among our Kentucky loggers that he had worked with the forestry department when he was an entomology student, and he really kind of wanted to focus on loggers. And I said, I have no idea what a red meat allergy is. He says, well, if a tick bites you, it turns out that it will inject this carbohydrate molecule into your body. And this particular carbohydrate is found in all animals except humans and apes, but it's found on beef, pork, mutton, and venison, the red meat portion. And if someone, not all people, but some people eat that, or excuse me, get bitten by that tick and get that sugar, the carbohydrate in their body, then they develop this intense sensitivity to red meat. Every time they eat red meat, have a hamburger, have a steak, they can get gastrointestinal issues, can get intensely ill. They can even develop anaphylaxis. I said, no way, no such thing. Sure enough, he was right. And so the question was, for us, is among this population, are we seeing alpha-gal? And so we went to the master logger training and forestry, fishery, and wildlife meetings, and we recruited people. And so we had them do a questionnaire. Most of them had not heard of red meat allergy. And we asked them if we could take their blood, and we sent that to Dr. Scott Cummins' lab at UNC, who is probably one of our national leading authorities on this, and he would analyze their blood for antibodies to these. And it turns out that a sizable proportion, about a third of the people, would be classified as having antibodies, potentially sensitized at some point in time. They were bitten by a tick, they got the carbohydrate, and they elicited a reaction. And these were the main risk factors for that. Blood type B, it turns out, you have to have Scott explain the immunology, the biology to you. But it turns out blood type B is fairly protective for being sensitized to alpha-gal. If you're a person who's typically atopic, you have a history of hives and being allergic to things, excuse me, you got an increased risk for it. And then, as I was talking about earlier, if you're one of those people that got a tick bite and the reaction seemed to stay a little longer, maybe a week or two, then that's another indication that maybe you got the alpha-gal. Now, then Scott graduated. Scott's up in Michigan now, and he's got a great job with the health department up there. And we got that paper published on this. And since that time, we have been doing a lot more work on alpha-gal. It turns out that this is a fairly highly prevalent disease, particularly in our agriculture, forestry, and fishing sector. And like I said, 30 to 40% of our people had the IgE antibody. This was similar to a German study with people who either worked outdoors or spent a lot of time recreational in the woods. None of our people in our study reported knowing that they had any red meat allergies, but about a third of those people did say they had frequent heartburn and gastrointestinal issues, not necessarily potentially associated with, at least in their minds, with red meat. And the major risk factors I've kind of already talked about. Hey, I'm getting really close to that 45-minute mark, Michelle, and I didn't even plan this or practice this. I think it was just luck. I'm down to my last couple of slides, which really, I think you told me we have a lot of industrial hygienists on the call today. All right, so I maybe frightened you a little bit about ticks and told you all about the kinds of diseases they can carry, and hopefully gave you an appreciation that not a lot's known, and a lot of people don't know about this, and we probably need to be informing them about this. And it's growing. It's getting worse. We're seeing ticks come into areas that they were never in before. We're seeing the populations increase, and we're probably going to see the infectious agents increase. So hopefully you learned that. What does that have to? And you know, there's certain occupational groups that are at much greater risk for this. What do we do about it? Well, besides the education, we can vaccinate. We're not going to probably go out there and catch raccoons and possums and field mice and vaccinate them, but at least we can vaccinate in some cases our animals for some of these diseases. It makes economic sense. They can be infected and they can get sick like we can. So that would be one thing. So making sure that veterinarians and healthcare providers in our region and our extension specialists and people in our associations, like the Cattlemen's Association, the Grain Growers Association, these people are aware of this. And then, well, how do we prevent it? Yeah, I can tell you, and I know not all of you would like this answer, maybe, but permethrin impregnated clothing does work. I can't remember, I think it's about 0.5% permethrin on clothes. Does a very good job. Now, you remember those pictures I showed of Anna and her team out there? I did not wear permethrin impregnated clothing. I would, make sure my sleeves, my gloves and my sleeves are all tight, and my pant legs were in my shoes, and I put tape around my pant legs to keep ticks from crawling up my pant legs. And I didn't, tighten up here though. I didn't do anything like that. Anna and some of her team wore permethrin impregnated clothing, and they told me during the campaign they never saw a tick on them. Every night I would come home and I would have ticks on me, even though I was trying to be careful. I was out there looking for them, right? So it wasn't like I was trying to avoid them. And every night I found a tick. Most of the time they were the little nymph guys. They were the tiny ones, but they were there. And so I would think that that is an advantage. Now I was using DEET. DEET, I'm not sure how effective it is. I wouldn't say it doesn't have any effect, but some. And so personal protection is kind of where education and personal protection is kind of where we're at this point in time. And I think that's another area, of course, doing surveillance and looking for these diseases. The next question is, so what? What can we do about it? And the intervention and the prevention, that's a longer range plan for us. And certainly welcome partnerships and ideas on what to do there, because I think that's the very next phase is how do we stem these diseases that seem to be on the increase, particularly here in the Southeast and in the Midwest. And but doesn't mean that you all out west are immune. They're coming your way. With Michelle, I think at that point I'm going to stop and let you tell me if there's any questions or any discussion people would like to have.
Excellent. Thank you so much for sharing all of that information with us today. At this time, if you have questions, please enter them into the chat, and we'll do our best to get to as many questions as we can. The first question that we've had come through: are golf courses less of an issue than wild wooded areas?
Absolutely. Yeah, we have not focused much on recreational. I think I mentioned that German study. But yeah, people who, I guess in Europe, there's a lot of what they call, I've only done it once. It's pretty wild, with orienteering. People go out with a compass and a map, and they're trying to get from point. It's almost like a road rally, only they're running around in the woods. And tick issues with that. And Europe is a lot like us. It's tick-borne diseases, not as much the mosquito-borne diseases. Mosquito-borne diseases tend to be more in the warmer climates, closer to the equator. But Europe is very much like us, and tick-borne diseases are very common over there, like they are here. And certainly recreational activities that take people out into the brush and the woods. And if I'm a terrible golfer, I would probably be one of the greatest risks, because I spend most of my time in the woods and the brush looking for my ball.
Oh, that's a great point. Yeah, when you have to go look for your ball. Thank you so much. Another question, you mentioned farmers and farm workers. Are there any kinds of, I guess, types of farms or farmer environments that are more or less risk than others that you've observed?
Yeah, I mean, I would think if you're going to segregate farming, now farming in general, you're outside a lot, I would say probably if you're doing the animal confinement thing, maybe you would be a little less at risk, but ticks can get in your flocks, could get in your herds, but probably a little less. Now here in Kentucky, one of the things we brag about is east of the Mississippi River, and I'm relying on my extension specialist to tell me that We're the #1 cattle producing state east of the Mississippi River. Those of you from Florida or some of the other states may argue with me. I'm just going to tell you, quote that. And I work with the Cattlemen's Association and cattle farmers a lot. And we're not talking great big, wide, expansive pastures for the most part. Kentucky's a very hilly state. Where we have the nice big pastures is where we're putting our grain crops. And so farmers spend a lot of time in fact, I was just with my dad this weekend. We spent a lot of time, we had a cow have a calf in the woods. And so we spend a lot of time in the woods with our cattle. And yes, ticks are already out there. It's May and ticks are there. And so cattle farmers in particular, and people that would spend if you're a hay farmer, sure. If you're growing that kind of a crop, you're at greater risk than I hope I touched on the habitat a bit. Grass and woods is where the ticks like to live.
Thank you so much. And kind of continuing in terms of exposures and occupations, we do have a question about construction workers. Do you think that construction workers will be more exposed to tick-borne diseases?
I definitely do. In fact, that's one of the issues. There's been a number of papers that have talked about this. I didn't really allude to them in my talk, but there's a body of literature so think about this. Lexington, Kentucky, our population is exploding. We're one of the areas of Kentucky where there are a lot of jobs, fairly good jobs, high-paying jobs. We have a Toyota plant up in Georgetown, which is only about 10, 12 miles away. I heard they're putting in another line. They haven't told us what car they're going to make, and that's supposed to bring in thousands of more workers. Okay, they're going to need a place to live. Where are they going to go? They're going to buy some land, and they're going to go into that land that was always a pasture or a woods or something. And the construction workers are going to cut the trees and form the land. And no question, those people are going to be at higher pressure for ticks. And we're seeing it. I mean, as I said, there's been a number of papers that have said that we're encroaching on land that was formerly tick habitat. And you think they're going to go away? Not right away. So yeah, construction workers would be another. I mean, that's what I hope. Clearly I'm focusing on the ag sector, being an ag center director too. But I think this is it's a big tent. There's a lot of occupations out there that I think are going to be a greater risk for these diseases, other than just agriculture and forestry.
Thank you so much. We have another comment/question. Wildlife doesn't honor political boundaries. Do you know of any coordinations between county public health departments with the state or in between states? What kind of surveillance or multi-state federal level progress or initiatives are you aware of?
That's a good question. I'm going to mention one that's completely different here to kind of answer that question, which I think is a good story, but then maybe I shouldn't, but I'm gonna stray into not as good a story. The good story is, it turns out that in Kentucky, we have never had a case of raccoon variant rabies. That's a big deal, because in Virginia and North Carolina, raccoon variant rabies is rampant. And if you know about raccoons, they're industrious little guys, and they get in people's garbage, and they eat your dog food, and they get on your back porch. And so people are at greater risk for being exposed to a raccoon from a wildlife standpoint than most other wildlife. Like now in Kentucky, we have a lot of bat variant rabies. But in all honesty, most of us aren't going into bat habitat a lot. In fact, a lot of our caves have been declared off limits because we're protecting bats from white nose syndrome. But so basically, I'm not going to come in. My risk of coming in contact with a bat is not as great as raccoon. The states have been working to fly over the Appalachian Mountains with helicopters and drop a vaccine bait, so the raccoons eat this. Now I'm told, maybe somebody on the talk that knows way more about rabies than I do. I know relatively little, but And we did do a paper on raccoon variant rabies, and this seems to be pretty effective. This is a coordination with wildlife, forestry, fisheries, and wildlife, U.S. Department of Agriculture, the states of West Virginia, North Carolina, Virginia, Maryland, Tennessee, Kentucky, working on this problem. And to date, I've been in Kentucky now almost 14 years. We have still yet to have a raccoon variant rabies. So that shows how states can come up with a solution and work together to try and of course, you know, who's paying the bill? Well, it's mostly federal dollars, but probably Virginia and West Virginia and North Carolina, it's costing them more, but yet we're the beneficiaries of this. So that's a good thing. Where it's a little murky is if you've tried to coordinate things across state departments of public health, there are collaborations that are fabulous and there are collaborations that aren't. And so I'll leave it at that. Is here in the Southeast, we try to partner with virtually every state south of the Ohio River and east of the Mississippi River, and that's kind of for our ag center. That's our region, and we have had mixed success. Some states are great friends, and some states we just don't. I'm not saying they're mean. We just don't work with them as well as we do other states. I should say no more.
Thank you for sharing. We have another question. What are some of the data set sources, or if you have the best data source that you're aware of for tracking the emergence of illness cases?
CDC. I mean, that's the issue. Is it now? This is the nice thing. I mean, CDC, being an employee of them for 24 years, I'm not going to say anything bad about CDC. They fed my family for a couple of decades. Fabulous organization. I mean, we are so blessed in this country to have the Centers for Disease Control and the heroes that are working there, the things that they do for us and the data they provide. I mean, if I have a student, and I know all of you who are professors or deal with this, is how many times have a student come to you and say, I don't know what I want to do, I don't know anything. And then you can just get them on the CDC website. And, you know, those of us in occupational safety and health, get on the NIOSH, the OSHA, the MSHA websites, and boom, you got data. And so now the problem is of course, from an infectious disease standpoint, you know about the notifiable diseases, we rely on the good graces of our healthcare community. And I would say that probably we're tracking 100% of the rabies cases. If we get a rabies case in Kentucky, it's going to make news. But other diseases, not so much. I mean, I think we're probably vastly underreporting Lyme disease. I mentioned that. We vastly underreport COVID. We vastly underreport influenza. And so some diseases we just don't report. So having said that, still, I think the CDC website's the first place to start. Some states have outstanding surveillance, sub-surveillance systems, and some not as good. But CDC is where I would start. You want to know about these diseases. And the other thing about CDC, they have really nerdy people there who know everything in the world about Lyme disease. They know its genetic makeup. They know everything about it. So you can contact those people. They may take a while, but the CDC does have a system for getting back to you. And we've had workshops where we just had a workshop in our Southeast Center conference was in Tampa this year. We have a South On conference. I think those of you in occupation, you know, there's a West On. I think there's a Neon Northeast Center or something. We are the host for the Southeast conference. We partner with the other ERCs and ag centers. Our meeting was in Tampa, and we had some fabulous speakers. One from CDC came down, and she with a focus on vector-borne diseases among occupational groups. It was fabulous. So Maria DePerio was the speaker. She was great. And so I would encourage people to reach out to NIOSH and CDC for information and for guest speakers.
Thank you. A couple more questions, and then we'll wrap it up here today. We've gotten a few different questions around testing, both for Lyme disease and also regarding the red meat allergy. Some people struggling with false negatives or flexibility in the disease test interpretation, retesting, et cetera. Are you able to share what you know of, if there's been any advancements in disease detection for some of these vector-borne Lyme tick-related diseases, or is
Not
it?
being a lab person, I probably should defer to somebody else. I would argue there's no question. There's not only is there misdiagnoses, as I said, I did have a slide that tried to emphasize this. The tests are expensive. They're complicated. Not everybody can run them. I think every state does have, an infectious disease lab, and I would argue they're, for the most part, they're pretty good. But people aren't. It takes a while to get to the state, and so if you've got some symptoms, and particularly symptoms that feel like you got the flu, what's the old adage, and they tell you in medical school, when you hear hoofbeats, think horses, not zebras. The first thing the physician's going to think is not ehrlichiosis, right? It's not Rocky Mountain spotted fever, unless they see a very distinctive rash. And so, first of all, even ordering the test, but you're right, even when the tests are ordered, and I don't have off the top of my head the sensitivity and specificity rate for these tests, I would argue it's another area of research. And it's not going to be my area of research, but I would, the alpha-gal thing, I had to go to North Carolina, University of North Carolina. And I don't know about now, but back when Scott and I were doing this in 2016, we started this study. We had a hard time finding somebody who knew how to look for the antibodies for alpha-gal. And so I think this is getting better. More and more papers are coming out. I have to plead more ignorance, though. I don't know the answer to those questions.
Thank you. Our last question that we'll get to for today, in terms of education and training, what would you recommend to employers in high-risk injury or in industries, in terms of making materials available to their employees?
Well, again, I mean, I think this is our ERC in our region is doing that. I think I showed you my little baseball card idea. We have tracks that we put out. We refer people to CDC. The CDC website is fantastic. The CDC has outstanding presentations that you can get online. I think they do a good job. At one of the arguments I have, I think they've dumbed down some of their literature. I shouldn't say these things, but I think they've dumbed down some of their literature a little too much. I think that people are smarter than maybe they think they are, but I think CDC is a good place. Reading the scientific literature, there's papers coming out on this sort of thing all the time. And if in your region, I know there's people from all over the country, we now have 18 ERCs. Reach out to them. If they don't have somebody who's focused in this area, we are partners, and we're brothers and sisters here. So there are some of us who are focusing on this area. Be happy to share what we know, and as things develop, that's one of the cool things about being a member of the ERC family is we share this information with each other, and so it can go that way. So.
Amazing. Thank you so much.
And I see some points. There's a lot of really good websites. I said some of the states, they rival CDC. They are really good putting out information. An extension program. So we've been working with our ag extension, as I said, for our focus on agriculture, but construction and even transportation and some of these other areas. I think there's groups that can be looking at that too.
Awesome. Well, thank you so much for your time today. Thank you to everyone who joined us. We are out of time, but we appreciate you all being here. We appreciate the engagement and all the questions and reactions to the webinar. Thank you all so much for being here. This presentation was recorded, so we'll send you all a link to the recording. You'll get that tomorrow via email from Zoom. And thank you. You can also check out the website for more information. And be sure to check out those additional resources that Dr. Sanderson shared with us. Thank you, Dr. Sanderson. Thank you, everyone who joined us. Hope you have a wonderful rest of your day.
Thanks a lot. Bye-bye.